Welcome to this comprehensive guide on cell culture techniques. Let's start with a hands-on demonstration. Maintaining sterility is the foundation of cell culture. Begin by disinfecting all work surfaces and equipment with 70% ethanol. Always work under a laminar flow hood to protect your cells from contamination and ensure the air remains sterile. Prepare your growth medium by combining essential components. For Dolbeco's modified Eagle medium, a standard recipe includes 500 milliliters of Dolbeco's modified Eagle medium, 25 milliliters of fetal bovine serum, 5 milliliters of penicillin streptomycin. Mix gently to avoid foam formation and pre-warm the medium to 37 degrees Celsius before use. Thaw your cryopreserved cells quickly in a 37 degrees Celsius water bath to minimize stress. Gently transfer the contents to a 15 milliliter centrifuge tube containing pre-warmed medium. Centrifuge the tube at 300 G for 5 minutes. Once centrifugation is complete, carefully remove the supernatant containing cryoprotectants, such as dimethyl sulfoxide, using a sterile pipette. Resuspend the cell pellet in 10 milliliters of pre-warmed growth medium and transfer it to a sterile culture flask. Place your flask in an incubator set to 37 degrees Celsius with 5% carbon dioxide. This environment mimics physiological conditions, supporting cell growth and maintaining pH stability. Monitor your cultures daily for growth and contamination. Now, let's dive into the theoretical principles that underpin cell culture. Cell culture is a cornerstone technique in biomedical research. It involves isolating and maintaining cells in a controlled environment. Let's explore its key principles and applications. There are two main types of cells used in culture. Primary cells are directly extracted from tissues and closely mimic in vivo conditions, but their lifespan is limited. Immortalized cell lines are either modified or naturally adapted to proliferate indefinitely, offering consistency for long-term experiments. Cells require a supportive environment to thrive. This environment includes the culture medium, which supplies essential nutrients, growth factors, and maintains a stable pH. It also includes the incubator, which ensures the correct temperature and carbon dioxide levels for cell growth. Lastly, the sterile environment is necessary to prevent microbial contamination, ensuring the purity of the cultures. Common media used in cell culture include Dolbeco's modified Eagle medium, RPMI 1640, and minimum essential medium. These media are specifically tailored for different cell types to ensure optimal growth conditions. Selecting the appropriate culture flask and medium volume is crucial for maintaining healthy cells. The appropriate guidelines are based on the size of the flask. For a 25 square centimeters flask, you should use 5 to 7 milliliters of medium. For a 75 square centimeters flask, you should use 15 to 20 milliliters of medium. For a 175 square centimeters flask, you should use 35 to 50 milliliters of medium. It is important to ensure that the medium fully covers the cells, but leaves enough space for gas exchange, which is necessary to maintain a balanced pH and support proper growth. Cells follow a predictable growth pattern that includes 4 phases. In the lag phase, cells adapt to their new environment. In the log phase, cells rapidly proliferate. In the stationary phase, growth slows as nutrients become depleted. Finally, in the decline phase, the rate of cell death exceeds the rate of cell division. Subculturing is crucial during the log phase to maintain healthy cell growth. Cell culture is indispensable in several key areas. In drug discovery, cell culture is used to screen potential therapeutic compounds. In toxicology studies, cell culture helps assess the safety of various compounds. In cell-based therapies, cell culture plays an essential role in developing treatments such as chimeric antigen receptor T-cell therapy. Now that we've covered the theoretical principles, let's review common mistakes to avoid during cell culture. Contamination by bacteria, fungi, or mycoplasma can ruin experiments. Work aseptically and inspect cultures daily for signs of contamination, such as cloudiness or unusual colors. Overconfluent cultures can deplete nutrients and trigger cell death. subculture cells before they exceed 80 to 90% confluence. Improperly prepared medium can alter cell growth. Ensure correct pH, sterility, and supplementation with essential components.